Thank you for the kind introduction. Good morning, everyone. I'm Peter from the National University of Singapore. Today, I'll be discussing our in-house platform, Identify. We developed this platform during the pandemic to optimize actionable drug combinations with pandemic readiness. So when the pandemic first started, repurposed drug candidates like remdesivir were prioritized to treat COVID-19. However, as more drug candidates emerge, the treatment strategies shifted towards combination therapies for several reasons. They may improve efficacy, lower toxicity, and also prevent resistance. So there are some important strategies to develop drug combinations, including high throughput screening, synergy prediction, and even higher order interaction. But in order to be truly optimized, we need to pair the correct drugs at the right dose ratios. For example, if we want to assess 12 different drugs at 3 different dose ratios, we're looking at a parameter space of more than 500,000. It will be time consuming and labor intensive to screen all 530,000 combinations. So when the pandemic started, we developed this platform called Identify. It harnesses an AI-derived biological relationship. Using neural networks to determine the relationship between drug interventions and the measured biological response in vitro, we found that the relationship can be mapped out by a second-order quadratic series. This platform does not use any big data or any pre-existing information, and it only relies on a small set of prospectively obtained data. And it is readily adaptable to any indications like HIV, tuberculosis, as long as 2 factors are satisfied: a quantifiable input and a reliable biomarker or output. And it can rapidly interrogate the entire parameter space in a timeline between 2 to 3 weeks. Here in Singapore, we work with clinicians, infectious disease experts at the National Center for Infectious Diseases, and other scientists and engineers to come up with this Identify optimization workflow. It begins with clinical engagement. So we want to come up with a list of drugs that are clinically actionable and feasible, and drugs that clinicians are comfortable prescribing. So we've come up with a list of drugs that are actionable and clinically relevant. Subsequently, the Identify optimization prospectively tests a set of combinations, typically ranging from 50 to 150 combinations. And we correlate the data that we collect prospectively via a second-order quadratic relationship. Then we can come up with a ranked list of all possible combinations in the parameter space and prioritize the ones that are clinically actionable and relevant. And of course, we experimentally validate all the combinations to make sure that they are effective against a pathogen or viruses. So when the pandemic first started, we harnessed this platform to pinpoint actionable combinations against the original strain of SARS-CoV-2 virus, as well as the Delta and Beta variants. And one of the most important drugs that we found in the study is EIDD-1931, or molnupiravir, an orally available COVID-19 drug. This drug may potentially serve as a backbone for combination therapies against COVID. For example, in the figures, we have EIDD-1931 in combination with remdesivir. And we can see that for each of the variants, the interactions are different based on the response surface. And also at different dose ratios, we see different interactions. So it is important to not only pair the correct drugs and combinations, but also determine the optimal dose ratios. And later on, we also use the platform to find actionable combinations for Omicron. And similarly, EIDD-1931 showed up as one of the backbone drugs for combination therapies. And we can see that in the figure on the right-hand side is EIDD-1931 in combination with favipiravir. And again, at different dose ratio, we see completely different synergy and different interactions. So dose optimization is important aside from pairing the correct drugs and combinations. And recently, we have also been working on resurrecting failed therapies. For example, antibody therapies like sotrovimab have their emergency use authorization revoked by the FDA. So we want to design combinations based on these failed therapies to make them effective and potent against different pathogens. And aside from COVID, we have also been working on antimicrobial resistance. For example, in this study, we used the platform to address non-tuberculous mycobacteria. And the identified design combinations were comparable to the standard of care, thus providing alternative treatment options to the existing standard of care regimens. And the most important thing I want to point out in this slide is that the combinations highlighted in red are the combinatorial designs determined by identified to be non-effective against NTM. And they were in fact non-effective. So this platform may potentially accelerate AMR-specific drug development to provide insight into helpful combinatorial designs and the ones that are not effective. And in a recent study, we used the platform to address gram-negative bacteria like Klebsiella pneumoniae. And we pinpointed a combination that was broadly applicable to 3 different gram-negative bacteria and their clinical isolates. It's bleomycin in combination with meropenem. And what we have observed is that sometimes at lower doses, we see better effects, better inhibition against a bacteria. And also, because the combination is bleomycin in combination with meropenem, we replaced meropenem with other carbapenems. We also saw synergistic interactions across multiple strands of the 3 listed bacteria. So this platform may potentially contribute to future pandemic readiness for several reasons. It is readily adaptable. It doesn't have to be COVID or AMR. It can be readily adaptable to other disease indications. And it is a rapid workflow that can be done between 2 to 3 weeks. It could potentially be faster depending on the logistics. And it is a community effort. This entire workflow is a collaboration between engineers, scientists, clinicians, and infectious disease experts, and many other communities here in Singapore. And the most important lesson that we have learned from combinatorial drug therapy is that we have to pair the correct drugs and combinations and determine the optimal dose ratios in order to achieve the optimal outcomes. And aside from pandemic readiness, this is our most recent study and has nothing to do with infectious diseases, but it's on N-of-1 health optimization. We harness a suite of digital health devices to monitor a subject's biomarker dynamics with interventions like intermittent fasting, fitness regimen, including cardio workouts and strength workouts, as well as dietary regimens, mostly Mediterranean-based diet. And we were able to observe the subject's fasting-driven ketosis or metabolic switching, also known, which is when the subject's primary source of energy switched from glucose to fat. And by looking at the subject's biomarker dynamics, we're able to gamify adherence to healthy lifestyle. And at the end of the study, the subject lost around 7 kilograms of weight and with blood pressure lowered and stabilized towards the end of the study. And that's it for my presentation. Thank you all for listening to my presentation and my team and colleagues here.